Welcome, everybody. We're going to get started in several minutes, so you still have time to grab yourself something to drink, get comfortable. Today's webinar is going to be with Simulations Plus. All right, why don't we get started? Welcome, everybody. Hope you had a nice, long holiday weekend and you had nice weather. Today's webinar is with Simulations Plus, and it's the second of 4 in a webinar series. The title is Focus Your Obesity Pipeline for Clinical Success, Delivering the Right Compound at the Right Dose for the Right Patient. As usual, our agenda is we'll just briefly go over our rules of engagement. We'll give a brief industry perspective, and then we'll turn it over to Dr. Le Merdy from Simulations Plus, and he'll give his presentation, and then we'll have a Q&A session at the end. Put your questions or comments in either the chat or Q&A box. The difference is the chat everybody can see. The Q&A box only the panelists can see. We'll address all questions during the Q&A session. This is important. This is going to go on demand. So if you have to leave early for some reason, you can come back and watch the whole thing because it will be recorded. But your question will also get answered and you can see that. We'll also have a brief poll here after Max's talk. So some challenge areas when it comes to the clinical environment around metabolism and obesity and things like that is the biological complexity is one of the top ones. There's all types of moving pieces here, as everybody knows. The more we understand biology, obviously, the more successful and the better things are going to be. The clinical trial design, you can have high dropout rates, long duration trials, regulatory hurdles like cardiovascular issues, market access, insurers reluctant to pay without hard outcome data, manufacturing, again, going back to that biological complexity, becomes harder to manufacture biologics. And then portfolio strategy, that team that sits in a large pharma that all works together trying to get their particular portfolio out and successful. It's a very challenging environment, and it's very competitive. And then public perception, overcoming stigma around obesity medications and everything we see in the press and stuff. We just wrote a little article this week about all of these kind of all in one, and how important metabolic targets and work is overall in a clinical setting, and how each therapeutic area is actually most likely affected by this. So it's a very critical domain in the clinical space. So I'd like to introduce Max Le Merdy. He is the Director of Research and Collaborations at Simulations Plus, and they are a leader in modeling and simulation software for the pharmaceutical R&D industry. He earned his PharmD and a master's in pharmacometrics from the University of Paris, Descartes. Max joined the FDA as a postdoctoral fellow, focusing on PBPK models for locally acting drugs and publishing work on ocular delivery and pediatric pharmacokinetics. So with that, Max, I'd like to hand it over to you.
Thank you very much, John, for the introduction. And let me share my screen.
Looks good.
It worked. Awesome, awesome. So good morning, good afternoon, good evening, everyone, depending where you're in the world. It's a real pleasure to be here today to talk about the obesity topic. What I'm planning to do for the next half an hour or so is to discuss, first of all, give you very brief information about Simulation Plus, what we do and who we are. Then I will focus about obesity per se. What are the definition of obesity and what is the impact of obesity on drugs PK? And finally, for the last sections is how we can actually model the obesity effect on the PK, showing you 3 validation case study we've used, and then we'll wrap up the whole presentation and open the floor for Q&A and discussions. So first of all, about Simulation Plus. Simulation Plus is a company that was established in 1996 and is now a worldwide company with more than 200 souls working for it. And all of the employees of Simulation Plus are working in some shape and form towards computational modeling, trying to predict the next step for drug development. So to clarify that, we are divided into a business unit, and you can see on the slide, so the cheminformatic, the PBPK, the QSP, the CPP, LAI, and the MC, medical communication business unit. And all of those business units can really like take a lead and support either through specific software or through consulting or clients or customers to really support their drug development efforts. And in today's session, so I actually belong to the physiologically based pharmacokinetic BU. And I will talk about how we can use the software called GastroPlus that we have developed and started in 1996, how we can use GastroPlus to predict the effect of obesity on drugs PK, and how that can actually support your drug development efforts. Maybe waive some clinical trial, or better anticipate whether you need to adjust the dose or not into the obese population. So as I mentioned, we are a software company, and we are developing different softwares that can be used at the different stage of drug development. In blue at the bottom here, you can see like the typical confidence level in clinical performances of a drug when used a traditional development, starting with animal study, preclinical phase, and then going to clinical, regulatory, and so on. The idea by using a computational solution is to support and take whatever information you have or you don't have and that can be predicted to understand more about your compound and predict the next phase. So it starts very early on in discovery space using AI and machine learning cheminformatic tool, especially ADMET predictor. Very early on, you can start using GastroPlus or GPX to start answering lots of questions regarding first-in-human predictions, DDI, safety questions, what is the impact of the food intake on the drug PK, and also answering questions regarding special population, including obesity. And then when you get like phase 2, phase 3, you have the population PK, adaptive learning, etc. And if you want more information on this, I would just invite you to have a look at our website where you can find all of the answers. And you can also reach out to us and we'll be happy to have a meeting and discuss any questions you may have. A little bit of self promotions because I'm very happy to lead the team actually working on all of those collaborations that we have with the FDA. And just to demonstrate that the PPK model and the PPK field is an evolving field that is always getting more and more complex and that always needs more and more validation. But we are very happy to work with the FDA as one of the main collaborators to develop and validate PPK models. And you can see on the screen the different areas we are currently working on with them, whether it's for LAI, virtual bioequivalence, locally acting drugs in the GI tract, pulmonary absorption, oral cavity, dermal. And you can see like last year, actually in 2024, we have finalized 2 collaborations on dermal drug product and an ophthalmic drug product. And you can see the outcomes of this research in publications. I will have a link towards some of those publications at the end of the presentation. So as I said, if you have any questions or if you want more information about the company, please have a look at the website or reach out to me and I'll be happy to either answer the questions or direct you to the people that have the answers. Now, let's have a look at the obesity per se. And the first questions or information is, let's have a look at some facts and figures about obesity. Here on these slides, you get the obesity classification and how it's currently defined by the WHO. And you can see that the metrics that is being used to define obesity is the BMI or the body mass index. And I will use BMI quite a lot for the rest of the presentation. So BMI is a ratio between the weight and the height with a square. And you can see that if the BMI is above 30, a patient would be considered obese. And then there are different classes based on the BMI level. So class 1, which would be a moderate obesity for BMI between 30 and 34.9, a severe obesity between 35 and 39.9, and a morbid obesity for a BMI above 40. Why is obesity important and why is it a challenge? And you can see on the screen the percentage of subjects in the US population that are either overweight, obese, or severely obese. And you can see how, like, if we combine the overweight, obese, and severely obese in the later stage, so over the last 10 years, so from 2025 to probably like a larger number now, there are weight concerns for close to 60% or more than 60% of the population in the US, and it's true for men and women. You can see obesity for both men and women in 2018 represented around 40% of the population. And if we combine the severe obesity, we can add like 10 more percent. So 1 in 2 person in the US is concerned with this disease, which means that when we want to develop drugs for patients, either we want to develop drugs dedicated to treat the obesity disease, or we want to develop drugs for other diseases, but for patients that will have obesity as a comorbidity. And we need to understand the impact of obesity on the PK as well for the drugs not aiming to cure obesity, but it could be like for kidney dysfunction or for cancer or for any other disease. But because obesity can impact the PK, we also need to understand that. So just a quick picture to look at the success with obesity medication that have been achieved recently, and actually showing, looking at these pictures, we can see that it's not actually a recent effort. The first drugs aiming to cure obesity or help patients affected by obesity actually started in the 1930s, 1933, with the dinitrophenol, and it was approved by the FDA in 1933. And you can see on the screen and on this slide, like the number of drugs that have been approved or actually withdrawn from the market from 1933 to more recently, like 2023. It's common knowledge now, like in 2021, semaglutide has been approved and has been a real game changer to treat obesity, to treat the obesity. In 2023, the tirzepatide was also approved. So now we have drugs that have or approved on the US market, have demonstrated their efficacy, and are being used at large scales to really help support patients affected by obesity in their weight loss parcours. So that has been the last two drugs really approved over the last 5 years or so. But you can see that there is a massive obesity pipeline across the pharma industry. Of course, Novo Nordisk and Lilly or the big players that everybody knows and everybody heard in the media. But there are many other players that are willing or currently working on drugs to help in the fight against obesity. And on the slide, you can see like Pfizer, for example, Viking, Amgen, AstraZeneca, Boehringer, and many others. But as I said, predicting or understanding the impact of obesity on the drug PK goes beyond just these drugs that are here to cure or support patients in their weight loss. It actually works for any drugs that will be given to patients that are affected by obesity, and how we can already anticipate what the PK could be for these patients. So let's look at what I've called the obesity factor and how it is impacting the drug PK. And I really want to give a shout out to this publication that has been published late 2023 by Julien Gaudieu and Samuel Leguet. And disclaimer, I don't know these people, although they are from France. And you can probably hear I have a French accent. So I'm not trying to promote any French jobs here. But they have made this amazing review paper looking at the effect of obese on the pharmacokinetic. I really like that title because they say it's not only an increase in weight. So I would really encourage you to go read that paper. It's really interesting. But I've just extracted like the key information and figures from this paper. And first of all, when we look at the absorption, we can see that on the stomach, there is an increase in gastric emptying and therefore a decrease in gastric retention. There's also an effect on the stomach pH. On the intestine side, there is an impact on intestine motility, transit time, modification of the pH, modification of the CYP3A4 activity. On the liver side, an increase in hepatic blood flow, a decrease in CYP3A4 activity, and finally, as well, other changes on the gallbladder, which means that if we look at this, and if we have, for example, a weak base drug that is eliminated by the CYP3A4, we know a weak base would get dissolved into the stomach lumen and then probably precipitate as it's reached the duodenum. But at least the fact that we have a difference in gastric emptying it would have an impact on the weak-based dissolution rate. An elevated pH would also have an impact on dissolution, and it's possible that we don't see the same dissolution level between a lean subject and an obese patient. So just this modification could really impact the PK. As I've said, we have a drug that could be metabolized by the CYP3A4. If I decrease the activity, I can limit the first pass extraction into the gut and into the liver. So that can also have a huge impact on the overall drug absorption and the API fraction absorbed between a lean and an obese patient. So combining this change, you can see it's already very complex, and we are just at the absorption question. If we look at the distribution now, you can see on the screen like the theoretical proportion between the lean body weight and the fat mass as a proportion of the total body weight. And the total body weight cannot exceed 1. So when we have patients that are considered like the ideal weight, so BMI below 25, we can see that overall the fat mass represents close to 20% of the total body weight. When we are overweight, the fat mass would be anywhere between 0.2 and maybe 0.25, 28 of the total body weight. But as you get into that obese category based on the BMI, and as the severity of the obesity increase, you can see how the fat mass is in proportion being more and more present as part of total body weight. And why it is interesting, because based on the drug characteristics, based on its slot P, its overall lipophilicity, etc., the drug will distribute, if I oversimplify it, more into like the lipid phase of the body or into the water phase of the body, or the lean body weight. So as you increase the BMI, you are modifying the ratio between the lean body mass and the fat mass, and therefore you're affecting the drug distribution. So that combined with the absorption will already have a huge impact on the drug PK in the body. But it's not just that, because there are also changes regarding metabolism. Once again, coming from that publication, you can see here there is an increase in blood flow, there is a decrease in CYP3A4 expression level, an increase in 2D6, a potential increase in 2C9, and a decrease in 2C19. So you can see that based on the obesity, there is an impact on the metabolism. And it's not just the metabolism, it's also the excretion, where you can see overall that there is a reduction in renal drug elimination in obese patients. So combining all of these effects, I've actually sampled this sentence from the paper conclusion is like obesity is not a simple weight increase, as it is accompanied by many physiological changes. And I think that we can actually modify this sentence and tweak it and be a bit more impactful in a way that obesity profoundly impacts all stages of drug ADME. And this is something we need to acknowledge, because it's not just for, as I've said already, not just for drugs aiming to cure obesity, but it is for all drugs that the pharma industry is working on. When we have changes in all of the ADME, there is one tool that is really helpful and can really help to support to predict the PK of a drug. It's PBPK modeling. And PBPK model, what they do, and you can see an example of the structures of a PBPK model that we have in GPX. So PBPK model, they really, their structure is based on the physiology. And we have physical parameters for each of those compartments, as well as the blood flow, etc. What we can do is we can modify, keeping the same model architecture, but we can modify the physical parameters of the model to represent different stages of obesity. And by doing so, we can help to predict and maybe even personalize drug therapy in obese population. And this is what I'm going to do in the next slide, is to show you how we've used this PBPK model to predict actually the changes between lean subjects and obese patients, and really try to understand the differences or the quality differences. So, simulating obesity in GPX. As I've said already, obesity is a disease, and modeling disease state in GastroPlus is not a new adventure. We started building new physiology in 2017 when we have introduced the hepatic impairment, describing the effect of cirrhosis on the physiology and therefore on the PK. And we continued this effort in 2018 when we first included obesity into GastroPlus. We also included renal impairments. In 2022, we had increased the NAFL and NASH disease into GPX as well. And when we released the latest version of the software last year in GPX, we have all of those disease state physiologies included. So anything related to hepatic impairment, renal impairment, obesity, are in the software. And we are considering obesity in a similar manner. Like, yes, we can predict drugs PK for obese patients, but we can also predict the effect of obesity on any drugs. Like, this is kind of the idea. GPX is highly modifiable. So if you want to alter these physiologies, if you want to create your own physiology, you can. And also, if you want to reach out to us and want to work on specific physiologies or specific states, same, reach out to us and we'll be happy to discuss and see how we can work together. I'm gonna introduce you a couple of the changes that we've made when we are considering obesity. I'm not gonna go through all of them because I would need 5 hours, but just the most important one. And the most important one is really the body composition versus the BMI. I got this sentence from that review paper I was presenting a couple of slides ago. It is stated that for obese people, the volume and the mass of some organs or tissues, such as the brain, the spleen, and the lungs, they do not vary with the increase in the body weight. However, for other organs, such as adipose tissue and liver, there is a significant increase. What I've done is going into GPX, I have created, using the proprietary algorithm, I have created 3 physiologies for 3 virtual subjects with the same height, 180 centimeters. And for one of these subjects, I set the body weight to 75 kg, for one of them to 100 kg, and for the last one to 140 kg. The BMI goes from 24 to 32 to 45 kilogram per meter square. So we have a lean, like ideal body weight on the left, an obese stage 1 in the middle, and a morbid obese physiology on the right. First of all, you can see that the total body fat, it's increasing. We go from 23% to 30% to 40% as we increase the body weight, which makes sense because we are describing the physiology as I was showing a couple of slides before. Now, if we dive a bit more into the numbers and we look at the lung compartment, you can see that independently of the body weight, the lung compartment in milliliters is always the same volume. It's always close to 1100 milliliters or 1.1 liters. Where we see a difference is in the fraction mass. And sorry, my arrows on top of it, but we can see the fraction mass is decreasing from 0.015 to 0.012 and 0.008 here, the fraction mass. So the lungs have the same size, but in proportion, they represent a smaller and a smaller fraction of the total body weight. If we look now at the adipose, the muscle, and the liver, we see that their volume is actually increasing, but not all in the same manner. And especially if we look at the fraction mass of the adipose, where we're at 0.3 for the 75 kg virtual subjects, we go all the way to 0.38 and close to 0.5 for the morbid obese subjects, whereas the liver mass goes from 0.022 to 0.019 and 0.017. So the change is not as pronounced in the liver compared to the adipose. And all of these change have been made based on published information. And we have collected data regarding adipose volume in the body and fraction mass. We have integrated those, built some covariates into the model to make sure that when we create a physiology, not only we create a physiology that mimics, a typical human being, but also the changes on the body composition mediated by obesity or representative of the disease. And we are not simply scaling up all of the tissues, but we are doing it in a mechanistic manner. So at the end, the created physiology in silico represents a real obese virtual subject. So that was for the body composition. We have it as well for the CYP3A4. And I was telling you in the previous sections that the CYP3A4 expression level is decreasing with obesity. And you can see that in the slides. We go from, I'm just looking at the CYP3A4, but we go from 0.34 milligram per grams of tissue to 0.19 to 0.06 in the morbid obese patient. As I've said, there are many other changes that we've made. And same, you can find more information directly into the software. Or if you have any questions, feel free to reach out. Now, I'd like to show you how we have actually validated that model. And I'm going to talk about in total 12 case studies. I just try to organize them a little bit. So the first one is for midazolam. And I got this data from a publication, I think from 2022, where they built a population PK model of midazolam and its metabolite in actually a population that is interesting because it was both overweight and adolescent. So we have two covariates that we need to consider in the PPK model. At the same time, the obese and the adolescent covariate. In this study, you can see the patient demographic information where the gender ratio between female and male, overweight and obese, and it's mostly obese, the age range, the body weight range, the BMI range, etc., etc. And you can see at the bottom the data that I have digitized for midazolam and its primary metabolite, the hydroxy 1 midazolam. The good thing with midazolam is that we had developed a PBPK model, and we already published it, to predict the PK of midazolam in pregnant subjects. And I'm listing here the publication that we had. And in this publication, I just extracted the figures from that paper. I'm showing you how the model can really well capture the PK of midazolam following IV and PO administrations in healthy subjects. These subjects from these studies are 20 years old and have a body weight of 73 kg. And you see the model is doing a really good job. So what we've done is we have integrated that model and we took those defined demographic information and we've created a virtual population simulation in GPX, fixing the number of individuals to 100. And in order to create this population, we have defined the age, the body weight, the BMI, and the gender percentage based on the published information. Then I let the model run. And here were the results. You can see the hundreds simulations in the slides in obese adolescent. For midazolam on the left, for the 1-hydroxymidazolam on the right. And you can see that overall, the PBPK model really well capture the observed concentration for midazolam in its metabolite in overweight and obese adolescents. Another case study is for voriconazole. And I got the data from this publication looking at the steady state plasma PK of oral voriconazole in obese adults. In this study, they had only reported data in obese subject with a BMI of 41.6, as you can see right here. And we had data at steady state for 2 dosing regimen, 200 milligram and 300 milligram. Voriconazole is also an interesting case study because it is known that this drug auto-inhibits its own clearance. So we have, once again, two covariates. We have a drug-drug interaction covariate on top of the obesity covariates, and we need to combine both into the PBPK model. So voriconazole is one of the drugs that we have in our DDI database that you get when you get the software to predict DDI. So what I've done is I've took this model and I took the data for those 10 subjects in obese patients. I've also extracted data from a paper from the same group that looked at voriconazole PK, but this time in healthy subjects in orange. And they have received the drug in a somewhat comparable clinical protocol because those healthy subjects, they first received the drug in IV for a week and then PO for a week. So what I've done is for each subject, I've changed, first of all, the physiology. I have considered the auto-inhibition of the drug, and then I'm also matching the clinical design for each of them. I've put all of this information into GPX, and these are the results that we got. So in orange, we get the data observed and predicted in healthy subjects for both 200 and 300 milligram PO. And you can see that the model is able to describe the data relatively well here and here. And in blue, we have the data in obese population at 200 milligram and 300 milligram. And I've also included like some zoom out. So you can see like not only the overall mean profile is well captured, but also the shape of the profile that is different to the shape that we have in healthy subjects is also well captured by the model. That was true for the 200 milligram, and that was true for the 300 milligram. So from this paper, in the conclusion sentence, they were saying that the oral dosing of voriconazole, it should not be based on the total body weight, especially in obese patients. Otherwise, you would increase the dose in obese subjects, and that could have a risk of disproportional exposure and toxicity. The modeling results are showing exactly that. First of all, we can describe the observed data, but we can actually extend and see that at steady state right here, the obese exposure is somewhat comparable in obese subjects versus lean subject. And even when we have a different administration schedules, we could reconcile the observed data and get to the same conclusion to what they had during clinical trials. So that was a very interesting case study, and I was really happy to see that not only we could capture it, but also, once again, in the context of DDI, being able to capture that. The last case studies, I should say, here is for cefazolin and metronidazole. Here, I got the data from one paper. And the good thing in this paper is that they reported data. So they did a parallel study where they look at the PK of the drugs in both obese and lean patients, but they try to match the demographic information between the two subgroups. I'll get to that. So they did a study, and you can see that they had like 10 obese and 10 non-obese subjects. And you can see that for the, let me take the laser. For the age, it's somewhat match. For the height, it's somewhat match. Where it's really not match is on the weight, because we have a BMI of 26 for the non-obese group and 51.7 in the obese group, like a very significant BMI. They have reported data in square in non-obese subjects or lean subjects and in circles in obese subjects. For cefazolin, they have reported the total and bound concentration, but I've used the total in the simulation. Once again, for both of these drugs, we have published PBPK model that we use to validate the prediction in the pregnant population. And you can see on the screen the validation for cefazolin administered IV at 3 doses, and for metronidazole following IV and PO in healthy subjects. So we had the PBPK models, we had the data. So what we've done is taking these two PBPK models, one for cefazolin, one for metronidazole, we have done single simulations and created PBPK physiologies for lean and obese patients based on the published mean information that we had here for the age, the body weight, the BMI, and the gender. By doing so, we got those simulations here. And you can see that for both cefazolin and metronidazole, there is a decrease in Cmax in both observed and predicted, but also a decrease in the clearance shape. And you can see here, even here, like how those observed and predicted data are crossing each other at close to 5 hours. So we had very interesting results, but it was only 2 drugs, so probably not enough. But when we look at these drugs and when we calculate the observed compared to lean Cmax and AUC ratio, you can see that in blue, the observed value, and in green, the predicted value using the PBPK software, GPX. And you can see that for both cefazolin and metronidazole, the PBPK models could accurately predict the change in PK metrics between the lean and obese patients. We've actually done the same exercise for 8 more case studies, and you can see that overall, GastroPlus can predict the obesity-mediated changes in PK metrics really well. You can see, like for the 4 compounds for whom were either long IV infusion or PO administration, the Cmax decrease between between lean and obese is well described. You can see that for most drugs, either the decrease or increase in AUC is also well described. And probably the only case study where we see some conflicting information is for cimetidine, where we had 2 studies. And you can see here that one study show like no differences between obese and lean subjects. One study showed a significant effect, and the model was kind of in the middle, showing an effect, but not as drastic as the clinical data. But overall, what we see is like the observed and predicted are really close to each other, but also the directions, whether it's for the Cmax and for the AUC, is also always correct. When there is an increase, the model is predicting the increase. When there is a decrease, the model is also predicting a decrease in AUC. So that can really help you to predict what is the PK outcome for your population for your obese population. And once again, whether your drug is aiming to cure or support those obese patients in their weight loss journey, or whether your drug is aiming to cure any disease but you know there will be some obese patients in your patient population. So just to summarize, I think it's clear that obesity profoundly impacts all stages of drug ADME. Because the PBPK model structure is based on the human body physical architecture, its parameters can be adjusted to describe the physiology of obese patients. We started to work on obesity in 2018 and then optimized and improved the models. Overall, you can see that GastroPlus can really well predict the obesity-mediated changes in PK metrics. But also, interestingly, not just obesity, but at least in the case of midazolam and voriconazole, the model could do really good prediction of the obesity impact in the context of pediatrics and DDI. So combining those different covariates and comorbidities really becomes really interesting. So with that, I would invite you to visit our resource center for more information. And if some of you will be at AAPS, I will try to present some of that work at AAPS in San Antonio this year. And feel free to connect with us and reach out. Happy to take any questions, either in the next 10 minutes or so, or after the webinar. You can reach out to John, and I'm sure he's going to forward me the question, or happy to share my email.
There's just a quick poll here to see if folks would be interested in someone from Simulations Plus to follow up with you. We'll run it for like 30 seconds. All right, great. Awesome. Thank you. At least 50% of the folks are interested in a follow-up, so we'll make sure you get reached out to. Let's see. Let's go to the first question here. Yes, will the slides be available to us? Max, will you be able to provide slides to folks?
Sure, yes.
Okay.
I'll share them with you after the webinar, and you can probably share them with everybody.
Okay. Question around modalities, Max. Could you say what types of drug modalities are you guys working with your models? What's optimal? What's in progress, etc.?
Yeah, thank you very much for the question. It's really good. So on this slide, you can see kind of a summary of the work we've done on obesity here, at least I've been working on. And you can see that currently I've only talked about small molecules. And these small molecules had a log P between, I think, minus 0.5 and 2.7, the highest, or maybe 3. So interestingly, like we are continuing the validation effort, trying to look at more hydrophilic and more lipophilic drugs as well. So going to the log P of 4 to 5, and then maybe like -2, -3, depending. Also looking at large molecule MABs, trying to see if we see an impact on obesity on the PK of MABs. Maybe it's not as likely because the distribution typically remains to that extracellular compartment. And the absorption, I mean, so far they were given either IV or sub-Q, but it could be interesting to look at the sub-Q administration. So there are still some ongoing efforts here. And yeah, if you have some questions or specific questions regarding like specific entities, feel free to reach out, and I'm happy to discuss.
Great. Max, the next question is around data. If you're working with a large, obviously Simulations Plus has curated data for many years now. And I know from experience that can be a daunting task. Do you work with clients? In other words, are their models a closed box, or can you work with clients' data to even improve the models or outcome, et cetera? How would that work?
Sure. So as part of the business model, like we have a consulting group, and we have like a highly dedicated scientists working in the consulting area, where typically the way it works is like our scientists really like or forming a team with a company scientist where we go over all of the data available for a specific compound and really trying to develop the model for the client, but meeting on a regular basis to explain the model, explain how the data have been integrated, and what is the outcome. So it's not just, you send the data to us and you get a report, but it's really like a collaboration effort in a way where everything is learning as the project is evolving. So that's step one. Step two is you can also license the software if you wish, and you can really start doing the modeling on your own. And we have also a program like to kind of teach you and help you in the first phase to learn how the program works and how you can use it. And the step three is if you're working in the space of obesity and you have access to a significant data set that you think could help us to further validate the model, I'm actually in charge of all of the collaboration aspects. So you could reach out to me. Happy to have a discussion and see how we could maybe embark into a collaboration project to further extend the validation and work together on that. So lots of
All right.
options.
Yeah, no, that's awesome. Some options are good. Next question is, how was the PBPK model developed, as in model structure, input values, output results, etc.?
Ooh,
That's a
you want me to summarize 25 years of effort in 2 minutes?
oh, maybe
um,
it might have to be at that level, like, at a high level.
no, it's been very incremental. So for example, GastroPlus started with an oral absorption model and gathering data, make sure it works. And then we built like it was kind of building a Lego. And every time we added a Lego brick to our model, we made sure that it worked, it was connected. And in a way, like we can consider obesity as one of the model brick. And you can see that, and you can see on this slide like the validation and how it's working. But the approach was always the same, is collecting data into the public domain or working with clients that had data and were willing to share them, building the model, making sure it works, do the validation, like validate. And once it was fully validated, like that Lego brick was added to the model in the commercial platform.
Very cool. Next question is around safety, and are any of your models able to predict or warn of safety issues or situations? For instance, you talked about renal outcomes or results, how the kidneys can control blood pressure and are responsible for a lot of different homeostasis and things like that. Are you guys providing any sort of safety predictions or observations from any of your models?
So I'm going to give you a very French answer. It's going to be yes and no. The yes part is, for example, if you already have identified a toxic dose
Mm-hmm.
in specific tissues or specific cell lines, what you can do in the PPK model, and because of its architecture, if I go back to that slide, is what you can actually do is predicting the either single dose or multiple dose steady state overall exposure in a specific tissue, and then you can correlate that, like for example in the kidney, and then you can correlate that total exposure with known in vitro toxicology data and try to define whether there is a risk of side effects and so on. So that was the yes part of the answer. The no part of the answer is what we don't do when we do PPK model. We don't try to simulate too much on the toxicologic effect and how these tissues are talking to each other and what could be the consequences of a side effect on the kidney, on the heart, and so on. This is where quantitative system toxicology really takes the lead I think the next webinar of this series given by my colleague Scott Seiler, who is the chief scientist of the QSP and QST. And this is really where like those models are even more complex, typically like thousands of parameters all connected together, and really can really support to predict the side effect. And they have developed recently, and it's been available to everybody, what they call the obesity sim QSP models that can really help to predict some side effect as well as the weight loss of drugs targeted to like at least have an impact on the obese disease.
That's pretty great. I think the last question is around future forward-looking. Max, it's always sometimes the hardest one to answer, but where do you see this space going? What's some of the future roadmap, if you can say, or ideas that you have of where PBPK is going, where Simulations Plus is going, etc.
Actually, I'm going to take that voriconazole case study because I think it's giving you all of the answers that we need in one slide. Is you can see here that we had individual subject data. What we didn't have were the specific covariates. We didn't have their weight, individual weight. We didn't have their metabolic capacity and so on. So what I had to do is I had to model them as one group. I could have done the same as midazolam and create like a population simulation, but I would not have been able to really map a specific physiology or specific simulation to an individual subject. And I think PBPK has really all of the necessary tools right now and existing to go and really make a huge step forwards towards individualized medicine, because we have the model architecture. We know the parameters, we know the disease changes. For example, one thing we can do is, and actually, if I go back here, as I was saying, like we have an example of for cefazolin and metronidazole, like PPK models that have been validated and that can predict the PK in pregnant subjects. In those slides, I've shown that it can also predict the PK in obese subjects. So maybe, we can have, like have a model that would follow us during our lifetime and based on our current physiological state, predict what the dose should be, what the administration schedule should be, et cetera, et cetera. So a huge step towards individualized medicine is probably like, at least to me, like where things will go in the future.
Very cool. Well, believe it or not, we're at the top of the hour. I don't see any more questions coming in. Great work. From an efficiency perspective, what I can't imagine that companies uh, aren't, aren't working with you on these things to drive efficiency, especially in like clinical trial optimization, at that portfolio level, even trying to get more data before they put maybe their candidate forward or whatever. Do you see much competition out there with your clients? Not necessarily other software companies, but are many of the groups internal at the larger pharmas and stuff building all this themselves, or are they mostly working with the likes of you and Simulations Plus? Just curious how that is today.
Yeah, no, so I think for the PPK software, like we have more than 200 clients that are licensing the software globally,
Yeah,
and they are building their own models. So we are developing the tool, we are making sure the science is correct, making sure the platform is robust, and then we are providing that platform to our client, and then they can build their own models. We have some training programs to make sure, people get acquainted with the platform. But mostly, I would say, like our job is to make sure the product, like the software, is solid, validated the best it can be with available data, and then people have complete independence to build their model and use it as much as they want for as many compounds as they want. Only
right.
time is the issue.
Well, it's great work. It was really good to see a lot of good questions from the audience, and I guess we'll see you in the near future in an upcoming webinar or other work. So, thanks very much for the great presentation, and we'll talk soon.
Thank you very much. Have a
All
great day.
right.
Bye bye.
Thanks, Max. Bye.